coming back to the previous slides, and this will be the sequence logo you'll look at a lot when you're describing a sequence specificity of a transcription factor. But this type of presentation is kind of very straightforward. It's very simple. And it identifies identities of letters. So there's a big assumption here, which means, let me see, the identity of letter at a given position are independent of the identities of letters at the previous position, or the ones after that. Which means that this letter has nothing to do with this, this has nothing to do with this. And that is the assumption of this kind of type of model. But sometimes it's not always true. Probably it's never true, okay? And so there's an alternative presentation, which is a Markov chain, okay? I'm not going to get into it, but the point here is the previous, so the second letter is kind of dependent on the first letter. If the first letter is A, the second is G. If the first is C, the second has 20% to be G, 80% to be T, or things like that. So there are some kind of research effort to look for this probability model of transcription factor based on this Markov chain. So the only thing that is on top of the previous model we talk about is doesn't assume that independency between the different letters. Okay, now let's get back to the topic of ChIP-seq. All right, motif finding in ChIP-seq derived peaks. So there's two major strategies when we look for the ChIP-seq data and we want to study what transcription factor, what's the motif in those regions we identified. At this stage, we assume we already identified the regions, okay, based on the peak finders of some other algorithms. And there's two strategies. The first one is de novo motif finding. So basically, I know nothing about the sequence feature of the binding protein. ChIP-seq experiment identified, let's say, 1,000 enriched regions. Which motif is enriched in the peak region I identified? That's the de novo motif finding scenario. The second one is to search for the existing database, like a TransSpec database. Okay, and we'll get into for the second part in a lot more detail. Let me spend about 2 slides to talk about the first de novo motif finding. This is not really practical or have too much application on that, but let me get through that and tell you why. So the question here is, I have hundreds, sometimes thousands of DNA regions identified by ChIP-seq, and I know some common motifs are there, but how can I find who they are? Okay, and this data can be derived by ChIP or ChIP-seq experiment, or it can be based on a microarray experiment after clustering analysis. So this is the example of microarray experiment of several different conditions. So if you see some genes, they are always goes up and down together, and this we consider those as a cluster. And most people at this stage will try to look for their promoter regions and to see whether there's some common motifs in those regions or not. So this is one scenario. But for our scenario is these are the sequence regions we identified based on the ChIP-seq. And then we want to find out whether there's some enriched motifs in those regions. And it's not really an easy task to do if you look at the problem in this way. So this is are the ACGT, the sequences you identified from ChIP-seq. And the bold ones are the real binding sites. Okay? And if they are perfect binding sites, like AACCGT has to be straightforward that's a little bit easier, and people still can do it. But we know that for the motif finding, there's a degeneracy there. So for one location, if there's a 1 letter or 2 letter mismatch, it can still be there. And at that stage, using something simple really doesn't help too much. And I think up to 2005, there's a huge research area of this de novo motif finding. And this is a couple of algorithms that I'm listed here. And there's a review paper in 2005 in Nature Biotechnology. They designed some benchmark data and trying to use different algorithms to see whether I can find those. So of course, you can read this paper, which I read that before, and I already forget everything I wrote, right? But the point here is you want to be precaution that most of those algorithms were developed not aimed for the ChIP-seq level of data, because ChIP-seq we got thousands or sometimes 10,000 enriched regions, and for those they probably get up to a few hundred fragments, and that will be fine. It's not like their math doesn't work when you go to 10,000 regions. It will just take much longer time to do, and you have to take precaution on that. So I heard there's some effort that to work on some de novo algorithm that based on the ChIP-seq level of data generated. And I haven't seen too much publications, but I think if you found some, you look at some, do let me know, and I'm happy to read those. The reason I don't put too much on the de novo finding is there's a problem with de novo finding algorithm. The first one is the computation is really expensive, especially with thousands of regions. But more importantly, that once you find some sequence regions, what are you going to do with those? Right? You don't know which protein are binding there, and you don't know what protein, what is the cofactors. So you really don't have too much information to help you to design the further experiment. So a better approach to do this is in existing databases, searching for the existing database. Okay, let me give you a little bit of resource if you don't know that regarding the existing database, what the data will look like. So the position weight matrix, those matrices I just showed you, of over 700 binding sites are documented in Transfact database. And there's over 200 are in Jasper database. So I just went to the Jasper database last night and just take a look at it, just type in AP1. And this is one of the things they showed up. So these are the descriptions of this factor, and this is the sequencing logo, and this is the position weight matrix. Okay? And we have almost 1,000, over 1,000 transcription factors that this position matrix is available. Okay? So that is a huge amount of information that we can take advantage of when we do the analysis on the ChIP-seq data. So how do we do it? Just give you an example. I worked out this last night, and let's see, hope it still works. So you can see that for one binding site motif, with known binding sequences, one of it. Okay, we know we have 1,000 of those, but let's try this one by one. Let's focus on the first one first. And these are the fragments, the DNA fragments for the ChIP-enriched sample. And these are the fragments, the sequences that I randomly selected from somewhere in the genome. Well, is that possible to be a binding site? Yes, it's possible. Is that likely? No. So if I randomly pick some genomic locations, it has very low likelihood that there's a binding site in there. But you can see that this is a ChIP-enriched sample. This is random selected regions. So these are the part that I labeled as the real where the binding site is. But at this stage, I really don't know this information. So what I can do is remember that I have those way that I got a position weight matrix. I got a new sequence come in. I can calculate the matching score of that particular sequence, how well that match with my position weight matrix. Do you remember that, right? It was 10 minutes ago. And if you didn't sleep. And now what you can do is for each individual sequence strain, this can be a couple of hundred base pair, and then you just design a sliding window just from the very left to the very right and calculate a score. You've got many different scores, but you just report the highest matching score of this particular sequence matching with the position weight matrix I got. Let's say the first one is 8.3 because there is a binding sites in there. 7.9, 10.5, this is a strong binding sites. This one doesn't have any binding sites, and the score is 2.1. I'm just making it up, right? So on and so forth, you will be able to calculate a matching score for each individual fragments you detected based on the ChIP-seq. It's going to be calculation-wise, it's pretty heavy, but it's not that bad. You can do that pretty quickly. And for the random identified regions, just select regions, you will still sometimes you happens to see one of the sequence has a real binding size, so it's a pretty big score, and once in there, but it will be in a much smaller frequency. Okay? Next step, you design a cutoff. So these are so many different scores. Now, I say my cutoff is 1. Okay? Everything larger than 1, there's a binding size there. We know it's not. Let's just make it up. Everything larger than 1, I think there's a binding size. Smaller than 1, no binding size. Okay. Now, I'm going to see what's the percentage of the rays, the sequences in the random set that is larger than my cutoff score. So if the cutoff is 1, and all these are, and only this one will be ruled out. So 8 out of 9 here are larger than the cutoff I've designed. And for the positive sides, 9 out of 9 are larger than that. So if I calculate this with cutoff at 1, the FDR will be the percentage in the random divided by the percentage in the ChIP-enriched site. This is 0.88. Okay? So that's one way to calculate that. We know 1 is not a good cutoff. Let's try another cutoff. 7. Of course, you want to try more gradient ones, right, to make your figure a little bit better. But I'm just making it up. If I use 7 as my cutoff, so you can see that 2 of this doesn't make it, and 2 of this make it in the random size. And then when you calculate this FDR, this is 2 out of 9 made it, 7 out of 9 made it. And your FDR here is about 30%. So once you increase the cutoff value, the stringency of your detection, and then if this represents the real binding site, you will see the FDR further dropped based on the ChIP-enriched samples together with the random samples. That make sense? Okay. So this is based on one binding motif. Okay. Let me give you one example. This is published actually from our group in 2008, and the PI of this project is Dr. Harunaka Shatri in Cancer Center. So you can see that at that time it was the estrogen receptor alpha ChIP data, and we used MCF7 cells and AKT-induced MCF7 cells. Okay, so 2 different conditions. Now we did ChIP, okay, identify the regions, but this motif finding part really is not affected by which technology you used. And we used position weight matrix documented in TransFact database, search for the ERE. ERE is the binding site of estrogen receptor alpha. So we do have the position weight matrix there. And the number of sites with FDR less than 20%. So you can see that I'm adjusting the matching score cutoff here, right? Once I increase this cutting off score, and that FDR really dropped. So at this stage, this will be my cutoff I will select to decide whether this is a binding site or not. And we can see that in wild type and AKT samples, we detected so many different ERE binding sites, and actually 13,000, hundreds of them are overlapping between 2 experiments. Okay? So this is kind of a confirmation that based on this strategy, because I'm targeting on estrogen receptor alpha, I'm supposed to see this ERE binding sites. But we didn't really look for only ERE, right? We searched for all the 762 candidates in the TransFact database. And then these are the ones that will eventually drop down to FDR less than 2%. And these will also be the other binding sites that we concluded that are enriched in the regions that we identified. And once we look for the sequence identities of these binding sites, they have a certain level of overlapping. So very simple strategy. Everything just based on what I talk about today. Nothing fancy, right? Based on the position weight matrix, you got 1 enriched regions, got those sequences, calculate the matching score, adjust your threshold, and to see whether your enriched region will beat the random selected region. That's all. Very simple strategy. But this is can help us to get to there. There's one more thing that I want to mention here is there is some kind of considerations when you select random regions. Okay, so there's one part that this is a chip-enriched regions. This is kind of straightforward. Whatever chip color, peak color you identified, I'm fine with it. And then and you just search your motifs in this region. And the second one here is it requires a little bit consideration. And we did something wrong initially. That's why that I noticed. Initially, we randomly selected some regions from just genome-wide. It's anywhere it's not near to the gene and any place, and I pick out those regions. And we initially find many different motifs, not only the ones that I listed there. And once I look, I start to really doubt about it. This is not possible, but their scores are really looking good, right? And then when I further look at that, I found there's a lot of those binding sites are CG enriched, okay? And we know that in the promoter regions, there's a lot of CPG islands, meaning those promoter regions, they are CG enriched. So if I'm comparing the chip identified region versus a random genomic region, okay, I'm ending up selecting the differences which potentially are the difference between the promoter region and the random region. It's not really anything to do with the chip experiment I did, right? So eventually we did adjustment when we published this paper is we have to pick the promoter sequence of the genes that are not targets as an active site. And of course, we did more selection on that, more careful than that. But general scenario is we pick those promoter regions as the negative sites, and then did this comparison, and then those meaningful binding sites start to show up. I'm going to stop here today, and for the last one, we will talk about it next time. The reason for that is we will have a quiz, and as suggested by some of your classmates, and this quiz will also be closed booked, okay? And so put down your notes and close your computers, and we'll get through this pretty quick.